Good morning, FS 330 students. I hope you had a great weekend and enjoyed some of the nice weather we had. Some announcements to start this week off. You have homework that opens on Wednesday and is due on Friday, April 17th. We're quickly approaching the last week of class. So we have this week and one more. Exam 2 has been graded. It is complete, but I am going to go through and review them. It's a little bit more difficult to review answers and make sure everyone is graded fairly on Moodle, and that's taking a little more time than I thought. I apologize for that, but I want to make sure everything is equal across the board. So a little bit more review on that, and we'll have the final grades. Remaining, we have extra credit, so don't forget that chocolate homework next week that's due on Friday, opens on Wednesday, due on Friday of next week. And then a virtual NC State dairy tour. So that is, you just watch this video of the dairy and write a 250 word summary. All you have to do is submit it and you'll get that, I think it's 3 points for that one and 3 points for the chocolate homework. Total of 6 points available. Those points will be added to your lowest exam score. So if exam 1 was the lower of the two, exam 1 and 2 scores, those points will be added to that particular test grade. All right, so that virtual dairy tour opens on the 17th, and that's next Monday, and is due on the 24th. All right, okay. And don't forget about your comprehensive final exam. If you guys have any questions about this, it opens Wednesday the 29th and then closes May 1st, which is a Friday. So be sure you get that done within that time frame. Again, 40% of the exam will be new material. So from dairy all through the different types of leavenings and breads, chocolate, and ice cream. And then 60% of the test will be comprehensive. So it can be anything throughout the entire semester. As we've gone through the semester, you've seen how the material has built upon itself, how colloidal systems keeps coming up, and supersaturation, starch gelatinization, all of these things that we've talked about all build onto one another in these more complex food products that we're talking about. So keep that in mind when you're thinking about comprehensive questions. That's the type of thing we're going to be looking at with those comprehensive questions. And you get to pick 3 of the 5. If you answer all 5 questions, I'm only going to grade the first 3 that come up. So it is pick 3 questions. Don't answer 2 that you don't want to answer or you don't feel like you would have a stronger answer for. So if you guys have any questions about that, we can talk about it on Wednesday as well. All right, so this week is probably one of my favorite weeks in the semester, where we start really talking about yeast bread and about flour. And flour to me is one of my favorite compounds. So I've already told you that sugar or sucrose is, because you can take that simple compound, right? Very small molecular weight compound, and just by different concentrations of it, heating it up, you can create different flavors. If you start to caramelize that sugar, it creates a totally different flavor profile. But if you have different concentrations of that crystal or that solute, you can get a range of different textures. And then further, if you change the macro structure of it from amorphous to crystalline, that gives you an additional structure. Okay, so it's amazing to me that you can do all of that with one very simple, small molecular weight compound. Flour is a little bit different, but it still has a lot of the same properties of that sucrose as far as in food systems that makes it incredibly interesting to me. Flour is pretty basic, right? You've got some proteins in there, definitely some carbohydrates with starch. Sometimes you might have a little bit of lipid, depending on if you're dealing with whole wheat or white flour. So depending on those things, just varying the amount of yeast, the amount of salt, the different type of microbe composition that's there, and the different grains, you can get a tremendous amount of variation. Anything from a baguette to a ciabatta, you don't have to have a whole lot of crazy ingredients to make bread. Just some salt, some yeast, water, and flour, right? And you can make a plethora of different things. Now, of course, you can start adding in more ingredients like milk or sugar, butter, other types of fat. and we can change the composition of this thing entirely, right? So we can get yeasty, soft dinner rolls that have a real buttery, almost caramelized flavor to them. Or you can get something that's very firm, like a baguette, very chewy, lots of big air pockets in the crumb that has a depth of flavor that is more than just flour. You can really taste the fermentation in a really good baguette. Something that has had a pre-ferment and had a lot of fermentation time, you're tasting that fermentation not as much as the flour, right? So we'll talk about all of these things today and really try to touch on what makes yeast bread so interesting and so diverse. The first thing I want to talk about is gluten development. So far this semester, we've talked about quick breads where we don't really want gluten development, right? We don't want these things to develop a ton of gluten and become tough. Anytime you have gluten development, it's going to toughen the structure of the product. It also gives it a lot of elasticity, which in yeast bread is key. That's how we're holding on to the CO2 that is produced in the fermentation process, right? Because our leavening is going to be the fermentation process. Those yeast are going to produce carbon dioxide, right? Just like you learned in your lesson, that's where our leavening comes from. So this gluten development is essential. In some breads, we want a high gluten development. In others, we want a more moderate gluten development. And it really depends on what you're doing with it. Now, how do we get gluten to develop? We've talked about this a little bit. Just simply hydrating flour, adding some water will get you there, right? Gluten starts to develop immediately. It won't develop a lot, right? It's still not going to be very weak if it's just hydration that you're doing. But with yeast breads, we really develop that gluten by adding a lot of shear into it, by kneading. You've seen several of the videos on the lessons and posted on Noodle. All of those videos have one thing in common. They're kneading the dough. They're kneading the dough or they're mixing it in a mixer. Either one accomplishes the same thing. We're getting very strong gluten development. So how is this happening molecularly? Gluten development comes from the combination of two groups of proteins in flour, glutenin and gliadin. These are the two different groups. Now you'll notice that the glutenin proteins here are these longer chains of proteins, right? Long chains of proteins. There's about 40 to 60 different proteins that make up this group. They're not soluble in water, salt solution, alcohol, not very soluble at all. They do have a high degree of sulfhydryl bonding. So the only other protein groups that we've talked about where disulfide bonding really comes in, and remember, that is that covalent bonding. The only other group that we've talked about that has a high degree of disulfide bonds in gel formation that really impacts texture is egg white, right? So we know creating these disulfide bonds really creates a very elastic, strong gel network, right? So these disulfide bonds are formed between the ends of the glutenin proteins. At the end of the long chain of protein, there's a sulfhydryl group, and it can bond to another long chain of proteins, right? And so we get this very extensive network. The glutenin also has big hydrophobic patches on it. Okay, so these are the patches that don't particularly care to interact with water. And these glutenins are really responsible for this strength and elasticity of the dough. So how much can we stretch it? Now, where does this other group of proteins called gliadins come in? Gliadins are made up of a smaller group, so 15 different proteins make this up. They are soluble in alcohol, but not water or salt solution, right? So think about what makes up a dough. Water, flour, yeast, and salt, right? So these two groups of proteins are not soluble in either of those. In fact, if you want to do a really neat experiment, take some flour and hydrate it until it's soft and malleable, knead it to really develop that gluten, and then put it in a colander under running water. And you'll start to notice that all of this white starch begins to wash out of it. But if you have a strong gluten network developed, you'll be left with pure gluten in that ball of dough after you've washed all the starch out. And it is very much like a rubber band. Okay, you can pull it and stretches, it bounces right back. It's quite strong. And we can do that because these proteins, glutenins and gliadins, are not water soluble. Okay, so this allows them to preferentially react with each other rather than the water, and makes a very, very strong network. So the gliadins are these little balls here that you see. They're little tight globs of protein. And this is responsible for the more viscous properties of these doughs. So when we talk about viscous properties, we're really talking about how something flows. So think about water. Water isn't very viscous, and if you pour it out on the counter, it'll flow everywhere. Now let's think about something that's thicker, maybe something like ketchup. You pour ketchup out on the counter, and it'll spread slightly, but not quite as much, right? So viscous properties are talking about the flow properties of a dough or any type of fluid, okay? So the glutenin is responsible for that elasticity, that ability to bounce back, and the strength of the dough, while the gliadin interacts with the glutenin, as you see in this picture. It's interacting with those hydrophobic patches on the glutenin. Okay, so gliadin is interacting with the hydrophobic patches on the glutenin, and it acts like a little ball bearing to allow the glutenin fibers to roll past each other. Okay, so that's where you get this viscous property. How do we develop gluten? It's really pretty simple. We need to add in some kind of shear energy. And the first thing you do is hydrate everything. So these two proteins can come together in this way, where the glutenin is interacting with another glutenin protein here at the end through disulfide bonding, very strong bonds. And then the gliadin proteins here are interacting with those hydrophobic patches on the glutenin, and they can roll past each other. So hydration gets these things interacting with each other. Kneading is really going to organize your dough, right? So it's going to organize this glutenin structure so that it's all lining up together uniformly in a given way. And that really strengthens the dough by encouraging interactions between these glutenin and gliadin compounds. Now, kneading isn't the only way. You can do this through folding. And we see that with the puff pastry, with the laminated dough, where we're rolling the dough and continually having to do this 8-10 times over a time span of several hours, in between which we're refrigerating. So this rolling of the dough is where our gluten is being developed. In fact, each rolling becomes more and more difficult because that glutenin is developing more, or the gluten is developing more and more. Now, fermentation can do this also. You may have heard of no-knead breads, right, where we don't need to do that kneading procedure of pushing the dough and folding it, pushing and folding, pushing and folding. This works because the fermentation is actually doing the kneading for you. Now, is the gluten development that you get through a no-knead bread the same as a bread that you've kneaded? No. But you can use this no-knead principle to make bread with a whole lot less effort, and I think that's why it became quite popular recently. Right? So the fermentation process produces carbon dioxide. And as that carbon dioxide is produced, it stretches those gluten interactions and gluten network that's formed. Okay? And then periodically you come in and you turn the dough and fold it over onto itself. And that sort of gets all that CO2 out, which is a byproduct, which is toxic to the yeast. So you want to release it to continue fermentation. So you fold the dough. And that helps align everything a little bit. Not going to develop as much gluten, but definitely has a part in gluten development. So if we add more things to the gluten network, how are we altering that network, right? One of the main things that we add to almost every bread is salt. If you have someone in your family or that you know that has high blood pressure, salt is one of the main sources of sodium chloride in their diet. Bread is one of the main sources of sodium chloride in their diet, which does have an effect on blood pressure. And salt is not just in bread for flavor. That is a good portion of why it is there. It definitely enhances the flavor. But it also has a great impact on the gluten network that's formed. It actually increases the strength of that gluten network through electrostatic shielding, through charge shielding. Right? So we can shield some of these charges on the glutenin proteins, and that will allow them to come together and interact more. right? Because we don't have so much repulsive charge on them. So this is encouraging electrostatic interactions. When we add salt, we shield some of those positive charges, negative charges there, because rate salt has positive sodium and negative chloride. And so we're shielding some of those charges and allowing those glutenin proteins to come together and be stronger. You can make bread without salt. You just will never get the gluten development that you'll get when you add salt. Okay? The other thing that we often see if you go to the bread counter and you're looking at different breads is acid. And this is usually in the form of a sourdough type of bread. This bread has a low pH and it has a lot more acid production because the fermentation of this dough is, which we'll talk about in just a minute, is both done from lactic acid bacteria and yeast, and usually takes a bit longer. Usually this is a much longer fermentation. So this low pH makes amino acids have a similar charge, and this increases the charge repulsion, right? So we're actually weakening that gluten network because of the acid that's there. So sourdoughs are usually not quite as elastic a dough to deal with as, say, a normal yeasted bread. So sourdoughs, let's talk a little bit more about this. This could be considered a pre-ferment, okay? Right now in my refrigerator, I have a sourdough starter, and it's been in there, I think, for about 3 years. And I just periodically pull it out, feed it a little bit more flour and water, let it ferment for a little while, maybe use some of it to make bread or pancakes or waffles, and then put it back in. So the way this fermentation works is not only do we have yeast to produce the CO2 and leaven the product, but we also have lactic acid bacteria, which are, as we know, the bacteria that ferment dairy products as well. Okay, so it's the same group of bacteria. These are the same group of bacteria that help ferment pickles and sauerkraut. They're all over the place, right? And this is no different. They live on the flour. So this mix of yeast and lactic acid is there already on the flour. So if you want to harvest your own fermentation or sourdough starter that's regional to your area, you just get some flour and you hydrate it and you slowly help it to grow and until you've increased the number of yeast that are there, and lactic acid bacteria, to the point where it can leaven bread, right? So these have a lot of regional qualities. I did post a video on Moodle that was talking about a sourdough library. I believe it's in the Netherlands, or Belgium, excuse me. And so this is a library of all these different sourdoughs. Some of them are from China, some of them are from San Francisco. San Francisco has a very characteristic sourdough bread that it is known for. So there's tons of different types of starters. They all behave differently because their yeast and lactic acid bacteria are very different, what makes up those two things. So these breads, of course, are more sour and have a very high acid content. And because of that, they have a much longer shelf life. That acid that's present, that lower pH, really prevents mold from growing on the bread for a longer period of time. It also decreases the starch retrogradation. So once we bake the bread, the starch granules swollen up and leached out some amylose. And as we know, bread staling is due to the retrogradation, the reforming of a network with those amylose polymers. And so having this lower pH actually reduces that network formation between those amylose polymers. But on the other hand, even though it lasts for longer, we have a much weaker gluten network. So that's one reason. One reason we have a weaker gluten network is this acidity, right? The higher amount of hydrogen ions in solution really participates in electrostatic interactions and weakens those interactions between the proteins, which is exactly what we want if we want gluten development. The other portion of this equation that weakens the gluten network is the fact that these bacteria that are present, the lactic acid bacteria, actually produce protease compounds. Now, these are enzymes that break up protein, right? So if these protease compounds are really active, these protease enzymes, they will go into that gluten network and start hydrolyzing, breaking apart that network, right? As that happens, it weakens the gluten even further. So in order to prevent this from becoming the overriding portion of this dough, we utilize salt. When these lactic acid bacteria are in a high salt environment, their protease production rate is much lower. So we don't have to worry quite as much about that enzymatic breakdown of our gluten network when we use a lot of salt. So consequently, sourdough breads usually do have a fairly high salt concentration. All right, so we've talked a lot about gluten development and what happens with gluten development. Keep in mind in the lesson there was a big list of things. I encourage you to go through that list of things that are added to breads, yeast breads particularly, that have an effect on that gluten development. Because if we are enriching a dough, right, we have unenriched and enriched doughs. Both of these are present in our lab that we would have done and that you are watching tons of videos about. So if we have added ingredients in there, it's going to affect the gluten network. Lots of sugar, like you may have added for, say, a cinnamon roll recipe, right? The bread has a lot of sweetener in it, not only just applied to it, but inside the dough itself. That sugar content decreases the protein concentration, right? So if we have less of these things to interact overall within the recipe, we'll have less gluten development. And then the thing that you have heard over and over again that I know you know by heart by now is when we add lipids to any kind of protein network, those lipids are going to tenderize it, right? And they do that by interacting with the hydrophobic portions of the proteins and preventing protein-protein interaction. Now we know that glutenin and gliadin have both extensive hydrophobic patches on them, especially the glutenin, right? So this is where your weakening of that gluten network is going to come into play through hydrophobic bonding of lipids. So go through that list, make sure you understand how different ingredients being added is affecting this gluten network. Now the next thing we need to talk about is Baker's math. And specifically, I want to focus on hydration. So Baker's math is a unique way to look at percentages in recipes and gives a baker a much better idea of how that dough is going to behave. It's actually easier for a baker to scale a recipe using Baker's math, okay? So let's talk about this, but we're going to start with what we typically consider as a weight percentage of ingredients, right? So all percentages are based on, wait a minute, did we skip one? I'm sorry, these are backwards, I apologize. So weight percentage of ingredients is going to be based on the total weight of the recipe. So here in this table we have the ingredients that are in the recipe, the weight of those ingredients, and here on the bottom, this is the total weight. So we have 650 grams of flour. The total weight of everything, flour, water, salt, yeast, is 1,165 grams. Now the way that we get the weight percentage is we take the weight of the ingredient and we put it over the total recipe weight multiplied by 100. So in this recipe, if we were just looking at weight percentage, we have 55.8% flour, 42.9% water, 0.7% salt, and 0.6% yeast. These all total to 100. Okay, that's your typical weight percentage. So if we want to talk about Baker's math, this is all going to be based on the weight of flour, the total weight of flour versus the weight of the recipe ingredients, all of the ingredients, right? So if we look at percent flour, that's always going to be 100%. 650 grams of flour over 650 grams of flour times 100, 100%. Now notice these percentages are not going to add up to 100%. These are really more ratios, okay? So we're ratioing everything to the weight of flour. So let's think about water. We have the percentage of water, also in the baking community or field talked about as hydration of the dough. So we have 500 grams of water over 650 grams of flour. Remember, these Baker's percentages are based on the total weight of flour. And so this gives us a hydration of 76.9%. This is actually pretty high for a dough. Now the same type of math is going to be used to calculate the baker's percentage of salt and the baker's percentage of yeast. So here we would have 8 grams of salt over 650 grams of flour times 100. 1.2%, and 7 grams of yeast over 650 grams of flour times 100. That's going to give us 1.1%. And these are pretty typical ratios of salt and yeast. Usually salt ranges between 1 and 2% in a recipe. Baker's percentage, not total weight percent, baker's percentage. So 1 to 2% of the total weight of flour. And yeast is usually somewhere in that range as well. So these are baker's percentages, and it gives you a better idea of what's there. So a baker can look at that type of recipe and say, oh, I know that this dough is going to be very slack and difficult to handle. I would like to increase the yeast concentration based on my experience with breads in my bakery. Okay, so let's think about the amount of hydration, because this is what I really want you to focus on here. If we have 57% hydration or less, this is going to be a really, really stiff dough. Very, very tight crumb with little tiny holes. Think bagels or pretzels. Very, very tight crumb. Okay. The more water you add, the more hydration of that flour there is, the more large holes you're going to get. Okay. The possibility for large holes is there. So if we're between 57 and 65%, this is still a pretty firm dough. We're going to get uniform medium holes. Think about a white bread. Okay, so these breads are typically enriched a little bit, but this is the type of, of structure you're going to get. Very uniform distribution of size and relatively small. If we have something that we start to get more hydration, 65 to 70% or even 70 to 80, these are very soft doughs. Okay, so you get much larger hole formation there throughout the crumb. The holes are not all the same size. You might have some big ones and some small ones, right? And this is very typical of a baguette. Ciabatta is even wetter. It would definitely be on the high end of that scale. And then focaccia is the same. As far as very, very slack dough, you can't really form it into a shape. You bake it in a pan, right? And you typically get very large holes with this. That's typical of focaccia. If you watch the ciabatta video, you'll notice that there's not a whole lot of formation. Ciabatta is another word for slipper, and that's what that dough is supposed to be shaped like. Very long and flat. Okay, and you'll notice when they're working with that dough, they're basically just kind of forming and shaping it, not pressing like with a baguette. They're just kind of manipulating it to be the right shape rather than a lot of rolling and pressing and forming. Right? And that's because of that high percentage of hydration. So when we go to make our dough, we have several different types of yeast that we can choose from. There's 3 main ones. Fresh cake or compressed yeast, which is typically only used in bakeries that are making bread actively every day of the week. Active dry yeast, which you can find on the majority of grocery store shelves, either in little packets or in a jar. And then instant dry yeast, which is now becoming more and more available. I believe I've seen this at Harris Teeter. You used to only be able to get it online, but now it is as available as active dry yeast. So what are the differences between these 3 different types? The fresh caked yeast, which you see here, is a moist product. It has about a 2-week shelf life in the refrigerator. So if you're not using this actively, it's going to die before you can get it all used up. And this one is alive and ready for action. So that yeast is as active in there. It is growing. You just need to add it to a matrix of food and water for itself, and it will continue to grow. So this is a product that is taken. The yeast are fermented in a large tank to increase in number. To do this, they need a lot of oxygen, by the way. So yeast growth requires a lot of oxygen. Yeast fermentation, like yeast multiplication so that you have more and more yeast cells, but yeast growth, the cells themselves, just surviving and growing, don't need a tremendous amount of oxygen. Okay, so the yeast from the fermentation tank where they've been bubbling oxygen through it, they get all of that solid material out and press it into a cake, just like you see here. Active dry yeast can keep on the shelf, so no refrigerated needed for several months, but it does go bad. But you have to revive it in warm water prior to use. So this is active dry yeast, and you can see here that it's granulated, right? Little balls there from the yeast tank. And so this is typically dried directly from the fermentation tank, spray dried into these little balls. Now, instant dry yeast is a whole nother game. You can keep this on the shelf for a year or more, especially if it's frozen. It lasts for a very long time. You don't need to revive it. You can directly add it to the flour, add the water, and start mixing. There's no need to do this step where you're adding the active dry yeast to a little bit of warm water, letting it come alive and start bubbling, and then adding that mixture into the flour. So with instant dry yeast, you just put it right into the flour, add the water, and start mixing. These are dried into porous rod shapes, and they hydrate really quickly. So you don't have to go through this reviving step. They're much more ready for action and alive than the active dry yeast. So let's talk a little bit about fermentation temperature. We've got our dough, we've added the yeast, the salt, the water, the flour, everything is all mixed together. So what we're looking for now is fermentation. You just need to let it set. One of the things that we can do to further manipulate the dough and control the fermentation is determine the temperature we would like to ferment at. Typically, if you have plenty of time, you're going to use a warm room temperature, so around 27 degrees. This is the best temperature for fast fermentation, fairly rapidly. If you don't have a dough that you want a whole lot of fermented flavor in, this is a great temperature to use. So for example, if you're making cinnamon rolls or pizza dough, you can use this warm room temperature because you're adding so much flavor from other ingredients, from butter and sugar and cinnamon, if this is cinnamon rolls, right? All of that sort of overrides the time or the fermented flavor that's present in the dough. And at 27 degrees, you're not going to get very many off flavors. Now, if you bump that temperature up to 35 degrees C, this is pretty warm, then this is going to ferment very rapidly. We have a lot of CO2 production of this, right? So our dough volume is going to increase very, very rapidly. However, we're sacrificing flavor. At 35 degrees C, we get a lot of off flavor production, a lot of acid production, along with that CO2 production. So keep that in mind. Now, if we have time and we have a dough where most of that fermentation is coming from, or most of that flavor is coming from fermentation, like a baguette or a ciabatta, we can use refrigeration to help us develop that flavor and slow down the fermentation rate, the yeast growth rate. So we can retard that dough in the fridge at cooler temperatures. And this will slow down that fermentation by about tenfold, right? So we can make up a dough at night, put it in the refrigerator, let it ferment all night long, bring it out, let it come to room temperature, and then shape that dough, put it back in the fridge, let it ferment for the majority of the day, take it out, let it come to room temperature, and then bake it, and we'll have a much longer fermentation period that generates a whole lot more flavors, like those fruity esters that we get from yeast fermentations, and a lot more acid-type flavors as well. So picking the temperature at which you want to ferment your dough is really important in how it impacts flavor. The last part of this is crust formation. Okay, so we've fermented our dough. Please watch all of the videos for forming that dough and slashing the dough for expansion. Okay, so all of that stuff is in the videos, and that's really more technique based. So let's talk about the crust, because the crust is really important for yeast breads. We don't want that crust to form before we've gotten full expansion of our loaf. Once we get crust formation, the shape and volume of that loaf is pretty much locked in. So we want to give that that loaf as much time to expand as we can. Now, the way professional bakers do this is they have ovens that have steam infusion at the very beginning. So they put the bread in, they turn the steam on for a few minutes, 30 seconds, a few minutes. It wets down the inside of that oven and gives a nice condensation layer on the dough. Now, this is done at the very beginning, right? And what we're doing is we're adding more moisture to the surface of the dough that needs to be evaporated before we can get crust formation. By wetting the surface of the dough, we give it more time to expand, okay, before that crust sits into shape. We're also doing something that's very similar to our two-step or two-stage frying for French fries. We're allowing the starch in the surface of this dough to gelatinize and for amylose to leak out. So if we do that, we get a much stiffer crust formation because we have that gelatinized starch layer on the surface. This is really kind of tough to replicate at home, and people have been trying to figure out the best way to do it, and everybody has their own little way. But just think about this as keeping the surface of the dough wet. One of the ways that I've found that works best for me in my kitchen is to spray the surface of that dough with water before I pop it into a really hot oven. Don't be afraid to use very high temperatures to bake your bread. You can start with that really high temperature, wet the surface of the dough down, and then lower it if it's getting a bit too brown for you. Okay? So by spraying the surface of the dough, we're essentially doing the same thing that the steam-infused oven did. We're wetting that surface, allowing it to have greater time for expansion because we can't form a crust until that surface dries out. It also gives the dough a better opportunity to have starch gelatinization on the surface. Okay. Another way that we can do this, especially for round loaves, is to use a Dutch oven. Remember those heavy red pots that we have in lab? That's a Dutch oven with made out of cast iron and with a cast iron lid. Very heavy, holds lots of heat. So you can put the bread in that Dutch oven, put the lid on it, and after about 15 or 20 minutes of baking, take the lid off. And you're essentially creating a steam environment here by putting the lid on. The moisture in the bread evaporates and creates a very steamy environment on the interior of that Dutch oven. And then that keeps the bread moist and allows that surface to expand, gelatinization to happen. You take the lid off, all that moisture is released, and now the crust starts to form. Okay? So 2 ways to give it a try at home and to enhance the crust formation. At the end of the day, what you really want is a very crunchy, crisp crust on the outside that has lots of Maillard browning that's taken place, that's developing flavor. Okay? So those are our steps in yeast bread making. On Wednesday, we're going to talk a little bit more about baker's math, look at some recipes, and try to figure out how the gluten network is changing based on the ingredients of that recipe, and a little bit more about pre-ferments as well. So I look forward to seeing you all on Wednesday. Come up with some questions. This is a very complex process to try to talk about without doing in the kitchen. I encourage you to get in the kitchen. If you have some flour, yeast, and salt, you got what you need. All right, we'll see you Wednesday.